embryology laboratory equipment and maintenance, routine maintenance and troubleshooting of equipment. In an embryology laboratory, maintaining the equipment used for gamete handling, embryo culture, and assisted reproductive techniques, ART is crucial for ensuring reliable results and successful outcomes. Regular maintenance, calibration, and troubleshooting of equipment minimize risk of failure, contamination, and inaccuracies. Here's an overview of routine maintenance and troubleshooting practices for common laboratory equipment. 1. Importance of routine maintenance. Routine maintenance is essential to ensure accuracy and reliability. Calibration ensures that instruments perform to their expected standards. Longevity. Proper care and regular service extend the lifespan of costly equipment. Contamination. Prevention. Cleaning and sterilization reduce the risk of cross-contamination between samples, reagents, and embryos. Regulatory compliance. Laboratories must adhere to safety and quality control regulations to meet industry standards, e.g., ISO, FDA. Second, routine maintenance of embryology laboratory equipment. A. Microscopes. Purpose: used for the detailed observation and manipulation of sperm, oocytes, and embryos. Routine maintenance. Cleaning. Regularly clean the lenses, stage, and eyepieces with a non-abrasive cloth and appropriate lens cleaner. Avoid using rough materials that can scratch the lens. Calibration. Ensure proper focus and alignment of the light source and objective lenses. Check for clarity, contrast, and sharpness. Condenser adjustment. Clean the condenser lens and adjust its position to ensure optimal lighting and clarity for embryo observation. Illumination. Replace bulbs or LEDs when they begin to dim to ensure optimal viewing of samples. Troubleshooting. Focusing issues. If images are not sharp, check if the objective lens is properly aligned or if the condenser is positioned correctly. Lighting problems. If the light source is dim or flickering, check the bulb, power supply, and light intensity controls. Distorted image. If the image appears distorted, inspect the objective lenses for dust or damage. B. Incubators. Purpose: incubators provide a stable, controlled environment for embryos by maintaining proper temperature, humidity, and gas levels, CO2 and O2. Routine maintenance cleaning. Wipe down the incubator's interior with disinfectant regularly to avoid contamination. Use non-toxic agents to clean surfaces that come in direct contact with embryos. Temperature and CO2 calibration. Check and recalibrate temperature and CO2 levels as per manufacturer recommendations. Ensure that sensors are accurate to maintain optimal conditions for embryos. Water trays. Regularly check water trays to ensure proper humidity and to avoid the growth of mold or bacteria. Airflow maintenance. Ensure air circulation is not obstructed by media containers or equipment inside the incubator. Troubleshooting temperature fluctuations. If the incubator is not maintaining the desired temperature, check the thermostat or heating element. Replace damaged components as needed. CO2 levels. If CO2 levels are fluctuating, check the CO2 tank or connections. ensure the regulator is functioning and properly calibrated. Humidity issues. If the incubator is too dry or too humid, ensure the water tray is full and positioned correctly. Check for any air leaks around the seals or doors. C. Centrifuges. Purpose. Centrifuges are used to separate sperm from seminal plasma, isolate cellular components, or prepare samples for further analysis. Routine maintenance. Cleaning. After each use, clean the rotor, buckets, and inside of the centrifuge chamber with a mild disinfectant. Rotor maintenance. Inspect rotors regularly for signs of wear, corrosion, or cracks. Tighten any loose components to prevent imbalance during operation. Balancing. Always balance centrifuge tubes of equal weight across from one another to avoid excessive wear and vibrations. Lubrication. Ensure that the motor and moving parts are properly lubricated according to the manufacturer's instructions to avoid wear on bearings and other components. Troubleshooting vibrations. Excessive vibrations may indicate an unbalanced load. Ensure that tubes are properly balanced and the rotor is not damaged. Unusual noises. Grinding or unusual sounds can indicate mechanical issues such as a misaligned rotor, malfunctioning motor, or insufficient lubrication. Speed or timer malfunctions. If the centrifuge fails to reach the correct speed or time, check the speed setting dial, timer mechanism, and power connections. D. Laminar flow hood. Purpose. Provides a sterile working environment for handling gametes, embryos, and biological samples. Routine maintenance. Cleaning. Clean the interior surfaces, workbench, and airflow components with ethanol, 70%, or another appropriate disinfectant after each use. HEPA filter. Replacement. HEPA filters should be replaced at regular intervals, typically annually or as recommended by the manufacturer, to maintain proper airflow and contamination prevention. UV light maintenance. Ensure that UV lamps, if present, are functioning and cleaned periodically. Replace bulbs when they begin to dim. Airflow monitoring. Check airflow velocity regularly with an anemometer to ensure it meets the manufacturer's specifications. Troubleshooting airflow issues. If airflow is weak or inconsistent, check for blockages in the filter, ductwork, or vents. Ensure the HEPA filter is in good condition and replaced as necessary. UV light malfunctions. If UV lights are flickering or not functioning, replace the bulb or check the power connections. Inconsistent sterility. If contamination is observed in the sterile workspace, check that the filter is functioning properly and the cabinet is being used correctly. E. ICSI machine, intracytoplasmic sperm injection. Purpose. Used to inject a single sperm directly into an egg during IVF treatment, typically when there is male factor infertility. Routine maintenance sterilization. All equipment in contact with gametes, such as micromanipulators, holding pipettes, and injection pipettes, must be sterilized between uses. Calibration. Regularly calibrate the micromanipulators for precise movement to ensure accurate sperm injection. Microscope. Check, inspect the microscope and focus system for optimal imaging quality. Air supply. If using an air pressure system to manipulate cells, ensure it is properly maintained and calibrated. Troubleshooting precision issues. If the micromanipulators are not responding accurately, check for issues with the mechanical components or calibration. Image clarity. Problems. If the images are blurry, check the microscope's objective lenses, light source, and focal alignment. Failure to inject. If sperm is not being injected, check the integrity of the pipette, the sperm injection setup, and the air supply to the injection system. 3. General troubleshooting tips for laboratory equipment. Monitor environmental factors. Keep track of ambient temperature and humidity in the lab, as environmental changes can affect the performance of sensitive equipment. Use manufacturer's guidelines. Always refer to the user manual or manufacturer's instructions for specific troubleshooting advice, maintenance schedules, and part replacements. Document maintenance and repairs. Keep detailed logs of maintenance activities, calibrations, and any issues encountered to track equipment performance and identify recurring problems. Staff training. Ensure that all laboratory personnel are trained to handle equipment properly and know how to troubleshoot common issues. Fourth conclusion. Routine maintenance and troubleshooting are essential for ensuring that laboratory equipment functions optimally. By regularly inspecting, cleaning, and calibrating instruments such as microscopes, incubators, centrifuges, laminar flow hoods, and ICSI machines, laboratories can ensure reliable results, reduce the risk of contamination, and extend the life of valuable equipment.